Good evening to you all. Let me see by hand if you can hear my voice and you can see my screen. Okay, great. I think a couple of you have shown that you can hear me and you can see my screen. Welcome back to SCH 214, Intermediary Metabolism and Biomembrane. We're continuing today with enzyme kinetics and inhibition. And I think we did something on enzyme kinetics inhibition. Even maybe when you were in first year, you did something on proteins. I didn't teach you SCT 106. I don't remember. Or did I? No. I think it's another lecturer that taught you also. So we continuing with enzyme kinetics, and from the beginning, we want to understand what enzyme kinetics is about. So the last time we did look at factors that affect enzyme activity when we were looking at the dependence of the rate of enzymatic reactions on temperature, on pH, on first of all on the concentration of enzyme, and also substrate concentration. So these were factors that we saw to be affecting enzyme activity. Now, what we mean by enzyme kinetics is that when you keep all factors constant and vary only one of the factors. So for instance, you can keep, say, enzyme concentration constant, substrate concentration constant, temperature constant, and vary only pH to see the dependence of the rate of the reaction on pH. And you can do same for all. You can even keep all these factors that I've mentioned constant and vary time. So for instance, you want to see how the rate of the reaction depends on time. That does it go up? Say, does the reaction increase with time? Obviously, with time, you will see that the rate of reaction from the beginning will increase up to a certain point, and then from that point, that rate of the reaction may begin to slow down, calm down, because of course products will begin to be formed and there will not be reactants present in the reaction mixture for the reaction to proceed. So as a matter of fact, those curves that we saw when we saw the relationship between rate of reaction and enzyme concentration, you saw a linear graph. That is a kinetic curve. So all the curves you saw, the dependence of substrate concentration on rate of reaction, you saw a certain curve where there was in the beginning an increase in rate of reaction, and it got to a point, then you had the rate of the reaction being constant. The dependence on temperature, where you saw at low temperature, rate of reaction is decreased. At higher temperature also, rate of reaction is decreased, but there is an optimum temperature at which you see the maximum rate of reaction. You see a similar curve for pH. So all those curves that we saw were kinetic curves. So when you are studying the rate of reaction in response to changes in these experimental parameters, what you are doing simply is kinetic studies. So you are studying the kinetics of say, an enzymatic reaction. So that is all what enzyme kinetics is about. But let's take a certain historical background of this. How did this come about? So, in fact, the first person who represented the rate of reaction depending on, say, substrate concentration mathematically with some mathematical model was Victor Henry. Victor Henry is French on paper, but biologically he is Russian. So under some family circumstances, Victor was supposed to be born in France and later be adopted by his parents in order for him to become a legitimate child, so that he will be received as, be recognized legally in imperial Russia as such. But that's a different story on its own, that whoever is interested in it can go and read more on it to see under what circumstances Victor had to be born in France and later, come back to imperial Russia. But he had all his, higher education in France, and so he was a chemist in France and got acquainted with some professors also in chemistry who were working in industry, working with enzymatic reactions in the brewery, and were interested in the representation of the dependence of reaction, say an enzymatic reaction on, say, substrate concentration, for instance. So they had these ideas, and they gave Victor some interest into such areas. So he began to study how the rate of an enzymatic reaction depends on say substrate concentration. And he was the first who came up with some mathematical representation and equation to represent the dependence of the rate of reaction on substrate concentration. But at the time when he brought that out, it wasn't very clear to the scientists of the time. I think it took over a decade or more for another scientist, that is Leonor Michaelis, together with Maud Leonora Menton. Can talk about Leonor Michaelis first. Leonor Michaelis was a Jewish German who, was first of all, even though he was interested in scientific research. Scientific research wasn't a thing that fetched a lot of income at the time, and so he decided to first of all study as a medical doctor. So first he was a physician, and then later he got into scientific research and had been a professor in some universities in Germany. Later he left to Japan, and from Japan he went to the United States. But while he was in Germany, also working on enzymatic reactions, working with the enzyme. Those times the enzyme that was mostly used in these studies was invertase. That's the enzyme. It's also known as a saccharase. The saccharase is just an enzyme that breaks down, say, sugar. The tabletop sugar. That's a disaccharide consisting of glucose and fructose. So just studying the activity of these enzymes. And Leonor, was studying this reaction with invertase, the breakdown of sucrose, tabletop sugar. And Leonora, she was a Canadian, and also at the time, women were not allowed to do scientific research in Canada. And she was so much interested in scientific research. She was actually one of the first women to become a medical doctor in Canada, and was very much interested in scientific research. But because at the time there wasn't opportunity for her to do that in Canada, so she decided to leave Canada and go to Germany. And so in Germany, she met Leonor. And in Germany, she worked together with Leonor to discover the representation of the substrate concentration, having effect on enzyme, the rate of enzyme reaction. So in their work, they actually were now able to more comprehensively work on what Victor had done much earlier and brought out what is referred to as the Michaelis-Menten constant, Michaelis-Menten equation. We will look at it. And since then, the Michaelis-Menten equation is what has been used universally the most to represent the rate of enzyme reaction based on substrate concentration. Even not only substrate concentration, but even enzyme concentration, even the enzyme concentration itself, substrate concentration, and different factors you can use the Michaelis-Menten kinetics to study that. There are others like Edie Hofstee, Lineweaver-Berg, but we won't go into those other ones. Whoever is interested can go and look at those things. We'll just, at this stage, we'll just focus on the Michaelis-Menten kinetics. So I think some of these stories are in a way motivating, so that you don't just get up and start studying some of these things and not knowing the background of it, how people actually worked hard in the past to come up with some of these things. And you see people traveling, moving because of their interest in working on something, want to bring out something. So for a woman in a country, Canada, that does not give her the opportunity, she decides to leave, go to a different place to go and do that. All that is I think is a motivating story that we need to listen to. At least you don't just sit and think that if things are not working for you around where you are, you just sit there. But make a move, do something. Well, Leonor, even at the end, moved from Germany, also went to the United States. And interestingly, she became a professor at the very last year when she was getting to retire. So that is another motivating one. If it was someone who say, what do I need to continue, improving myself, even if I get improved after I'm retiring. But she continued until she got to that level before she retired in the last year. Right, we can move on now to look at the Michaelis-Menten kinetics much more into detail. So just like we saw those curves in our last lecture, here you are seeing a representation of the dependence of concentration of different things, like concentration of substrates, concentration of product, concentration of enzyme, concentration of the enzyme substrate complex, all of these with time. So looking at time of a reaction, how do these concentrations, change over time? And Michael is mentioning they were able to study all of this and came up with this representation of how these concentrations change over time. So we can take a look at it much more, looking at the various stages of these changes in the concentrations. So if you take a critical look at it, let's look at first of all the black curve, the curve with a black line. That curve represents concentration of substrates. So you have in the beginning of the reaction, concentration of the substrate is high because the substrate is the compound on which the enzyme is acting. And so at the beginning, that substrate is going to be high in concentration because it is not yet reacting, it's not yet forming product. So the concentration is high. That is at time 0. Now you notice that there is what is being referred to as a pre-steady state, which is the green shaded region. And what do you notice in that green shaded region? You notice that there is, the blue dashed lines represent enzyme concentration. So the amount of enzyme which is present, that's a catalyst, a biocatalyst present in the reaction mixture is high. That is to say that is the maximum amount of the enzyme, or that's the amount of the enzyme that is taking part, or that is catalyzing the reaction. So if you have a reaction mixture that you have some substrate, let's say you have the sugar, and then you put amylase, you put a certain amount of amylase into that mixture, that sugar solution, to act on the sugar. So that's what it means, that you have some amount of substrate and you have some amount of enzyme. From the beginning, of course, that's time 0. And so those concentrations are high. Then you see that the enzyme concentration is reducing. I mean, reducing in the sense that some of the enzyme molecules will be binding to substrate to form the enzyme substrate complex. And that enzyme substrate complex is also represented by the brown curve. And so you notice that at the beginning, the concentration of enzyme substrate complex is increasing. But all these stages is being referred to as a pre-steady state. Why pre-steady state? See, the next stage to that is referred to as the steady state. Why steady state? Steady state because in that region, you notice that you have almost a constant concentration of the enzyme substrate complex. What does it mean? It means that at this stage, where it is shaded brown, you have all the enzyme molecules being bound to substrate molecules. So all the enzyme molecules are engaged. So that is a concentrated stage. It's a stage of concentration of the enzymes, concentrated with the substrate. That's what it means. So just like we saw the changes with substrate over time, you saw that there was a saturation curve. That's at a point where the rate of reaction, becomes constant, because no amount of substrate you introduce in the reaction again, no change will occur. You remember that? We saw that last time. Same thing is happening with the enzyme here, that once you have all the enzyme molecules being bound to substrate, then you have an enzyme substrate complex at a constant, concentration over a certain period of time. Similarly, since you have the enzyme molecules binding to the substrate molecules, you have the enzyme concentration also, reduced up to a stage where you see that the enzyme concentration is also constant. So that becomes a steady state. It's a state at which the rate of the reaction will be constant because all the enzyme molecules will be active. All are bound to substrates. They are all acting on substrate molecules at this point, so that the rate of the reaction at that point becomes constant, steady. Then gradually, you also notice that if you come back to the enzyme substrate, the substrate concentration, clearly that is logical because you see that the amount of the substrate in the solution is reducing because the substrate is being converted to product. And so concentration of the substrate continues to reduce all the way till the end, where now you don't have any substrates in solution. It's all being converted to product. And the product is being represented by the red curve. So that from the beginning, there's no product. But once the reaction begins, product is beginning to form. And so the concentration of the product in the solution begins to increase steadily all the way up. And so you see that you now have product at the end of the reaction. You have products, the enzymes that have acted on the substrate. Once all the enzyme substrate complex now gets converted, because remember the enzyme substrate complex is an intermediary in that reaction, and so it will eventually get converted to product, where the product dissociates away from the enzyme. And so you see that the concentration of the enzyme at the end of the reaction is equal to the concentration of the enzyme at the beginning of the reaction. Remember, the enzyme is only a catalyst. It's just catalyzing the reaction. Itself is not changing. So at the end of the reaction, you still have the enzymes now present free. And you have, of course, the enzyme substrate complex is zero because, of course, all of that enzyme substrate complex that get formed at the beginning of the reaction, they all get converted into products. Now, this was a more comprehensive representation of the kinetics of enzyme reactions by Michaelis and Maintain, which, of course, gave more understanding, more comprehension to scientists, more than what Henry represented the first time. But of course, they also depended on what Henry did earlier to be able to come to this. And so sometimes when you read some biochemistry books, they may refer to this not as Michaelis Menten. Some will even add the name of Henry and say Henry Michaelis Menten. Some scientists will maintain that Henry's name should be added to this because he was the first to, bring about the first presentation. Some will say a particular equation should be attributed to him, but all of that is just scientific discussions, people's opinions. Whatever it is, we do appreciate the works of these pioneers in this field. Right. Now, let's look at what is being referred to as the Michaelis-Menten equation. So, out of the representation of the rate of enzymatic reaction on these various factors which we just looked at, Michaelis and Menten were able to come up with an equation which relates the rate of reaction, say V null, to say the maximum rate of reaction, which is Vmax, and the maximum rate of reaction, that is the point at which the reaction, the rate of reaction does not change. That is, it becomes constant. That's the maximum you can reach. And the enzyme substrate, the substrate concentration. Now, they also came up with what they refer to as the Michaelis-Menten constant, which is represented here, Km. That's the substrate concentration at which half of the maximum rate of reaction is reached. Now, how do they derive all of this? So if you look at this curve, which we saw in our last lecture, that saturation curve of substrate concentration, depending on reaction rate, you see that if you take a tangent to this, say the very beginning going up, like if you just draw a straight line, and you remember your equation of straight line, y equals ax plus b, a is tangent or gradient, then of course you'll be able to derive this and come to this, but there's no point going through all of that. You can easily remember that the rate of reaction is at any point is equal to a maximum rate of the product of the maximum rates of the reaction and substrate concentration divided by the Michaelis-Menten constant plus the concentration of substrates. So, and then also you keep in mind what is the Michaelis-Menten constant, the Km. What is Michaelis-Menten constant? The Michaelis-Menten constant is the substrate concentration at which half of the maximum rate of reaction is reached. So if you see that this is the maximum rate of reaction, which is being shown up there with the dashed lines, and so the half of it, the corresponding substrate concentration on that curve is what becomes the Michaelis-Menten constant, Km. So these are the major things that you should remember. First, remember the Michaelis-Menten equation. And in the Michaelis-Menten equation, there is the Michaelis-Menten constant. Then, what is the Michaelis-Menten constant? Then you also remember that Michaelis-Menten constant. So these are two major things that you should remember from here, because these are the major things which you use to study enzyme activity. I mean, anywhere you go, if you are studying enzyme activity, you have a say some reaction mixture. So this is a practical thing. You have a reaction mixture that you put a certain substrate in there, you put a certain amount of enzyme in there, you know the amount of substrate you've put in, you know the amount of enzyme concentration that you have in there, and with time, if you run the reaction, use an instrument and measure. Perhaps when the reaction occurs, there is some change of color in a reaction, and so you can use an instrument like a spectrophotometer to measure, optical absorbance, for instance. And then you come up with a curve like this. And so from the curve, you will be able to determine the Michaelis-Menten constant. So it's a practical thing. You can determine the Michaelis-Menten constant after you have this curve drawn from your experiments. All right. Now, we can move on to enzyme inhibition. I think we did look at what inhibition was when we were looking at terminologies, common terminologies that we use in enzyme studies. We did see what enzyme inhibition was and what inhibitors were. Now, what we'll do is take a look at some common inhibitors that are applied as pharmaceutical agents, as medicines. And even think we'll look at least maybe one example of enzyme inhibition being applied in the production of, say, pesticides. In the control of pests, you have the application of enzyme inhibition there. And you do remember what enzyme inhibition was, as the reduction or the slowing down or prevention or stopping of an enzymatic reaction, and any compound, any substance that reduces the rate of reaction or stops the reaction or prevents a reaction from occurring, that substance is an inhibitor. And so once you know a substance that inhibits a certain enzyme, and of course that enzyme catalyzes a particular reaction, then of course you'll be able to make use of that inhibitor, that substance, to control that particular reaction in a biological system. So let's dive in and look at different types of enzyme inhibition. The first we are looking at is referred to as competitive inhibition. And what does it mean, competitive inhibition? Now you do remember we made reference to the active sites of an enzyme. The particular portion of the enzyme that interacts with the substrate and acts on the substrate. So here what we are seeing is the red structure is representing the enzyme, and the groove in it is representing the active site of that enzyme. And we see a substrate which is represented with the yellow or gold color. Another inhibitor here is it's also acting as a substrate to the enzyme. The inhibitor is also is represented with a green structure there. Now you notice that the substrate on which the enzyme acts, can fit into that active site for the enzyme to act on it. Now, if we take a close look at the representation of the competitive inhibitor and compare it to that of the normal substrate, you realize there is some resemblance between the inhibitor and the substrate. The inhibitor is, in this instance, usually referred to as a structural analog of the substrate. Structural in the sense that when you look at, let's say, the chemical structure of these substances, you compare them. When we go ahead, we'll look at this much more clearly. If we take a normal substrate and you take an inhibitor and you look at them, you see there's a resemblance in their structure. So that's why we refer to them as structural analogs. They are not functional analogs. Functional analogs will mean that they can carry out the same functions, but the inhibitor does not carry out the same function as the substrate, but structurally they look alike. And so it's some kind of if you look at it, roughly speaking, that the competitive inhibitor deceives the enzyme. The enzyme thinks that is its substrate and binds to it. But when it binds to it, it is not the substrate. And once it's not the substrate, the enzyme cannot act on it. So that's what is happening. And the word competitive means that you have the normal substrate and the inhibitor competing for the active site of the enzyme. Both of them can fit into the active site of the enzyme. So the inhibitor fits in. Once the inhibitor fits in, it blocks the substrate from binding to the active site, and so the reaction does not proceed. So that is how the competitive inhibitor will inhibit the reaction. Now, how do we represent this by the Michaelis-Menten plot, or the Michaelis-Menten kinetics that we just saw? Now you have here an inhibited reaction, which is represented with the blue curve. That's curve number 2. And you have the red curve, which is curve number 1, representing the normal enzymatic reaction, which is not inhibited. And so now, what do you notice? You notice that, if you take a look at the substrate concentrations against the rate of reaction, you notice that the rate of reaction at any point is lower for the inhibited reaction compared to that of the uninhibited reaction, the normal reaction. Now, you also notice that you have the Michaelis-Menten constant shifting to the right for the inhibited reaction. That is to say, the Michaelis-Menten constant increases in the inhibited reaction. That is in the case of competitive inhibition. Now, why is that? Now, another thing you should understand is that you see the word competitive is used. Both the inhibitor and the substrate can bind to the active site of the enzyme. Now, you can have a lot of times in competitive inhibition when you have an inhibitor binding to an enzyme. If you should introduce more substrate into the reaction mixture, that is increasing substrate concentration, that substrate can go to replace, it goes to displace the inhibitor, because it's a form of increasing the probability of the enzyme binding to the substrate molecule. So the more substrate molecules you have, the more there will be binding of the enzyme to the substrate. And so you would have displaced the inhibitor and you have substrate binding, and you then will have the reaction proceeding. I mean, what is referred to as reversible inhibition. We'll come to see that example. So the maximum rate of reaction can still be reached even with competitive inhibition. It's just that you will need more substrate in order to achieve the maximum rate of reaction. But the maximum rate of reaction can still be attained in the case of competitive inhibition, which means that the maximum rate of reaction is going to be the same, it's constant. That maximum rate of reaction can still be reached, only that it will be reached at a higher concentration of substrate. What it then means is that half of the maximum rate of the reaction will also be the same, it will not change. If the maximum rate of reaction is the same, is not going to be changed, then of course its half will also not change. Except that the concentration at which you reach that half of the maximum rate of reaction increases. So that becomes the apparent Michaelis-Menten constant, what you see here being represented Km.I, to represent that this Michaelis-Menten constant for the inhibited reaction. So here, the things to keep in mind is this. In competitive inhibition, maximum rate of reaction is constant. 2. In competitive inhibition, half of the maximum rate of reaction is constant. 3. In competitive inhibition, the Michaelis-Menten constant increases with inhibition. So these are three main things which you should keep in mind. Sometimes the last statement I made can be said that the Michaelis-Menten constant shifts to the right. That is to talk more pictorially. You are referring to the graph. So if you draw the chart, that's what you will see on the chart, that the Michaelis-Menten constant for the inhibited reaction shifts to the right. It's the same as saying that the Michaelis-Menten constant increases for competitive inhibition, compared to the uninhibited reaction. All right, so those are three key things that you should keep in mind. How about non-competitive inhibition? So in this case, if you look at the enzyme being represented with the green structure, you notice that apart from the active site, there is an extra site, which is not the active site, but that is a site that an inhibitor can bind to. You see, it's been when you reach, you come to say terms like allosteric sites. So that's site that you can have some substance compound binding to, but it is not an active site. Now, what do you see? Again, yes, normal substrate can bind to the active site. And you also see that after even you have that substrate binding to the active site, you have an inhibitor, that non-competitive inhibitor, binding to the other site, that allosteric site of the enzyme. So what it means is that there is no competition between the substrate and the inhibitor for that site. The inhibitor binds to its own site. There's a site there that the inhibitor can bind to. And here is being depicted in such a way for you to see that after you have a substrate binding to the active site, when the inhibitor binds to that enzyme substrate complex, it changes conformation of the enzyme active center there. So the enzyme no longer, fits to the substrate, and so the reaction is then inhibited. So you see that clearly with this diagram, that no competition is between the inhibitor and the substrate for the active site of the enzyme. Now let's take a look at the representation of this non-competitive inhibition, also by the Michaelis-Menten kinetics. How will this look like? Here again, we see the uninhibited reaction is represented by the red curve up there, the red curve, uninhibited reaction. And the blue curve is being represented by the blue curve is representing the non-competitively inhibited reaction. Now here, what do you notice? You notice that the maximum rate of reaction for the inhibited reaction is lower So the maximum rate of reaction reduces because, of course, the inhibitor is having its own site that it binds to. And once it binds to that site, even if you are introducing more substrate into the reaction mixture, that substrate cannot go to displace the inhibitor. And so the maximum rate of reaction is drastically reduced. And if the maximum rate of reaction is drastically reduced, then of course, the half of the maximum rate of reaction will also be reduced. And so you see it here. For the uninhibited reaction, you have maximum rate of reaction up there, written in red. And you have the half of the maximum rate of reaction for the inhibited reaction in blue. You see it below there. So there is that reduction of the maximum rate of reaction. Now, if you have the maximum rate of reaction reduced, as well as half of that maximum rate of reaction also reduced, what happens to the Michaelis-Menten constant? Now, the Michaelis-Menten constant remains the same. So the substrate concentration at which half of the maximum rate of reaction is reached is the same for non-competitive inhibition. So you see there, Michaelis-Menten constant, Km, is the same that at that concentration, at that substrate concentration, you reach half of the maximum rate of reaction for the non-competitive inhibition. And it is at the same substrate concentration that half of the maximum rate of reaction is reached for the inhibited reaction as well. All right. So these are the major things here too you keep in mind. The Michaelis-Menten constant remains the same for inhibited reaction and uninhibited reaction in the case of non-competitive inhibition. Similarly, you keep in mind that the maximum rate of reaction reduces for the inhibited reaction, just as the half of the maximum rate of reaction also reduces for the inhibited reaction. That's in the case of non-competitive inhibition. Now we do have inhibition also classified as reversible and irreversible inhibition. And this is also, very clear just from the words reversible and irreversible. I mean, reversible means that an inhibitor that binds to a substrate. So here is being depicted, you have a protein target. So that protein target, that is an enzyme, and an inhibitor is being represented by the orange structure. And you see the arrow is showing that the inhibitor can bind and unbind from that protein target. And so that's a reversible reaction. I think there are many, medications that function as, reversible inhibitors. They do not bind to a substrate and just, permanently, and that's it. They bind, they carry out their function, and then they can unbind. So you have that as reversible inhibition. Then you do have inhibitors that form covalent bonds, for instance, with proteins. That is, forming a permanent enzyme inhibitor complex that is not reversible. So that's what is being depicted below. You have a protein target, and you have an inhibitor, which is depicted with a green structure. And once it binds so tightly to the enzyme, it does not get unbound from that. It disables the enzyme, permanently disables the enzyme. We'll come to see some specific example of this. So now we begin to look at specific forms of inhibition. These are real applications of this, the use of these inhibitors as real applications in some medications. And then I think lastly, we'll look at one that is applied as a pesticide. So let's look at different, very practical, real practical, like it's being used as a, application of inhibitors. Now, when we started to study enzymes, I think at the point we did look at coenzymes, cofactors. And one of the coenzymes and cofactors that we looked at was the tetrahydrofolate, what we also refer to as folic acid. And you do remember that I mentioned that folic acid is essential for the development and proliferation of cells, which is why it's a lot of times prescribed for pregnant women because it helps in the development of the fetus. Because at that stage, the rate of the growth of cells and the proliferation of cells, specialization of cells into different tissues, becoming organs and all of that, the rate at which all that is occurring is very fast and requires that there should be adequate amount of folic acid. And so there shouldn't be any form of deficiency of folic acid at that stage. If that happens, that can affect what is being referred to as, say, maybe some mental retardation, because it results into what we refer to as defect in the development of neural tubes. That is to say defect in the development of the central nervous system in the brain and all the other, nerves accompanied with that. Now, in the synthesis of folic acid, what you see at the right-hand side is the process of synthesis of folic acid, or say the tetrahydrofolic acid. Now, this synthesis, it occurs in humans and it also occurs in bacteria. So you see, bacteria also requires folic acids, like the tetrahydrofolic acid, for its growth, say the growth of the cell, the growth of the bacterial cell. Now, we see that the synthesis of this, that is, so the bacterial cell actually synthesizes, it produces this folic acid, by itself, by, its metabolic reactions. The same way that, like say humans, we can also synthesize this folic acid, through our metabolic, reactions, the same way. Now, what do you see from the beginning? You see that there are two compounds. One is red, one is blue. They react, they bind together. So you have the dihydroterine pyrophosphate and 4-aminobenzoic acid, which sometimes is also referred to as para-aminobenzoic acid. Now, these two molecules react. You can see it clearly that they conjugate, they bind. Just by the removal of those two phosphate groups on that dihydroterine pyrophosphate, you see there is that conjugation, there is that binding between the two molecules. This is easier to see because the molecules are represented with different colors. So you can just see that the two of them just bind to form one, and they form dihydroteric acid. Now that dihydroteric acid then binds to glutamic acid. That's a green the green part that comes to bind to that. I mean, that glutamic acid, you don't know, that's an amino acid. So an amino acid, comes to get conjugated to that, first product there to now give you dihydrofolic acid. I mean, if you look at it even more critically, you can tell clearly that this is just like some peptide bond being formed. So it's a peptide-like bond being formed between glutamic acid, I mean, on its end terminus, that is the amine end of that glutamic acid that is binding to a carboxylic acid end of the dihydroteric acid. And so what it forms is a peptide-like bond. I say peptide-like because the dihydroteric acid is not an amino acid. If it was an amino acid binding to an amino acid, you can say it's a peptide bond. But because it is not an amino acid, but the bond is still between an amine group and a carboxyl acid group, and so we can say it's a peptide-like bond. It looks like a peptide bond, but it is not. All right. And so now you have that dihydrofolic acid. And the dihydrofolic acid undergoes some reduction reaction. I mean, if you just take a close look at it, you only see there are some slight differences between the dihydrofolic acid and the tetrahydrofolic acid. What you notice is, if you take the very first part, where you have the two rings, you see on this nitrogen, there is no hydrogen, and there is a double bond there. But when you come down to tetrahydrofolic acid, you realize that on that nitrogen, there is hydrogen. And a double bond is also removed. And so that's the difference you can quickly see and tell that you have some acceptance of some hydrides to remove that double bond from the molecule. And that is being catalyzed by dihydrofolate reductase. That is the enzyme that is catalyzing that last reaction. And the first reaction that we saw is being catalyzed by dihydroterate synthetase. So you are seeing these two enzymes catalyzing these reactions. And you also see that there is some small note being written beside the very first arrow, the reaction there. See, there's a cross on that arrow, and beside it, in that rectangle, is written sulfonamides. See, so sulfonamides are a class of antibiotics, and so it means that sulfonamides inhibit this reaction. They inhibit this first reaction where you have the dihydroterene pyrophosphate and para-aminobenzoic acid conjugating to give you the dihydroteric acid. Sulfonamides, that class of antibiotics, inhibit that reaction. And that's last reaction down there too. You see, same thing. You have this drug, this medication, trimethoprim. Trimethoprim also inhibits that last reaction, which means it inhibits this enzyme, dihydrofolate reductase. Same way, sulfonamides inhibit that enzyme, dihydroterate synthetase. That's what it means. We can take a closer look at each of these to see what this means. Now, this is an irreversible competitive inhibition. And clearly, what is the enzyme that is being inhibited? The dihydroterate synthetase. Now, what is the substrate that the inhibitor is competing with? The substrate is the para-aminobenzoic acid. And remember that I mentioned that the inhibitor is a structural analog of the substrate. Now let's take a look at the structures of these two. Say let's take one of the sulfonamides, a common one, simple sulfanyl amide, and take a look at the para-aminobenzoic acid or the 4-aminobenzoic acid. Now, if you take a close look at them, you see the structures look alike. The only difference is that the substrate, the para-aminobenzoic acid, is having a carboxylic acid group there. And in place of that, if you take the medicine, the antibiotic, sulfanylamide, it is having some sulfoamide group in place of that carboxylic acid group. So that's the only difference between the structures. Apart from that, everything else is the same. So these two structures look that much alike. So that the sulfonamide can bind to the active site of dihydroteresynthetase and prevent it from acting on para-aminobenzoic acid, as a substrate. So to say that you have the enzyme being blocked by sulfanylamide, so that you can no longer have para-aminobenzoic acid and dihydroteresynthetase pyrophosphate reaction being catalyzed to produce dihydroteric acid, that first reaction that we saw. Now, if that happens, then what is going to happen? You have the bacteria that requires this reaction to produce folic acid, which it needs for its growth. It means that bacterial growth is going to be inhibited. So this is an antibiotic that you can refer to as a bacteriostatic antibiotic. It inhibits the growth of the bacteria. It doesn't cause the cell, the death of the cell, but it results in inhibiting its growth. So that's a bacteriostatic substance. Now, the second, that last reaction also that we refer to, that you have a trimethoprim also acting as an inhibitor to dihydrofolate reductase. Now, in this case, trimethoprim is acting as a competitive inhibitor, competing with the dihydrofolic acid. But the only thing is, if you take a close look at trimethoprim, look at the structure, and look at the structure of dihydrofolic acid, you realize that the structures are not so much the same, but you notice if you take just the very first ring, there is a resemblance apart from some hydrogen, which is on dihydrofolate reductase, the first ring on that nitrogen. That hydrogen is not here. It's not in trimethoprim. In place of this amine group that you find in trimethoprim, if you look at the dihydrofolate reductase, you see there is a double bond, oxygen, that cyclic ketone group there. So that's different. And there is even not a fused ring. There is not a second ring which is fused to that ring. And then you have a totally different ring altogether. So you could see that it is actually, that first ring that can have interaction with the enzyme, which looks much alike like the dihydrofolate reductase. But this also brings to, the fact that, some antibiotics are more specific than others. So that's an an issue of specificity. That's, I mean, you see that the structures of some look much alike, like the substrate, and so they will be very, very much specific. But some of these, like broad spectrum antibiotics, will be acting on a large number of, antibiotics, a large number of bacteria. So you have trimethoprim also inhibiting that enzyme, dihydrofolate reductase, so that you have the prevention of tetrahydrofolic acid. And so once the bacteria is not able to produce tetrahydrofolic acid, its growth is inhibited. And so you find this antibiotic also acting in that way. I think it's one of these antibiotics which is used in some urinary infections and some others. Let's take a look at another one, which is much more specific. Methotrexate. Methotrexate is used even in chemotherapy. And you see that if you look at the structure, you can clearly see that methotrexate looks very, very, very much alike like dihydrofolic acid. So that medicine is going to be much more specific, because you see the differences there is maybe just methotrexate is having an amine group. That's when you look at the first ring. It's having an amine group up there. In place of it, there is an oxygen. And then you have the two nitrogens below for dihydrofolic acid. They are having hydrogen on them. Those nitrogens have hydrogen on them. That's not the case for methotrexate, no hydrogens. And because of that, you are having some double bonds in there, which you don't find in dihydrofolic acid. You have a methyl group on that nitrogen, where there is a conjugation, between the para-aminobenzoic acid and a terran ring. There's a methyl group. In place of that is a hydrogen atom there on that nitrogen for the dihydrofolic acid. So these are the differences in the two structures. And so this methotrexate is going to be, much more of a structural analog of dihydrofolic acid. And it also acts as an inhibitor to the enzyme dihydrofolate reductase. But you see, as I mentioned, that it's even used in treatment of cancer, which is to say that it goes to disrupt the proliferation of cancer cells. Because, the tetrahydrofolic acid, you see that is also found. I think you look at the biosynthetic pathway of the nucleic acids, you see that folic acid, acts, somewhere in the reactions, so that without it as a coenzyme, you have the disruption of the production of the nucleic acids. So that is a transfer of genetic material, so that you have the growth of cancer cells, the reproduction of cancer cells being inhibited. But again, since you have this reaction occurring also in normal cells, you do have, say in the case of chemotherapy, that is, a drug like this will be resulting into some side effects like hair loss, scaling of the skin, the nails becoming fragile, So yes, because even though cancer cells are are being targeted, in this case, it does happen that the drug can also go to affect like normal cells. That's why you have such side effects as well. All right. And so, of course, in the engineering of medicines, the more specific the drug is, the better. So that it doesn't have, or, the side effects are much, much more minimized. Okay. Now, before we look at another class of antibiotics, the penicillin, class of these antibiotics, I think we should remind ourselves here. I think we did look at when we were looking at mechanism of action of enzymes, we did look at a lysozyme that was acting on the structure, which is the main component of the cell walls of bacteria. You remember we mentioned that a peptidoglycan cell wall. Now that peptidoglycan is being shown much more here in detail that this is made up of N-acetylglucosamine. It's a derivative of monosaccharide, which is represented with this violet, the violet spheres. And you have the blue spheres, representing the N-acetylmuramic acid. You do remember we said in that structure, you have these two derivatives of monosaccharides alternating in that polysaccharide structure, and the polysaccharide, the oligosaccharide structure, and that oligosaccharide structure is linked with some peptide. It's a tetrapeptide that links this. So that's how come you are referring to it as a peptidoglycan. So peptido means there's some peptide component. Glycan is a carbohydrate. So you have the carbohydrate part is made up of this N-acetylglucosamine, N-acetylmuramic acid, and you have these dotted lines representing a tetrapeptide, which is cross-linking these oligosaccharides to give you that peptidoglycan layer of the cell walls of bacteria. So in fact, this is being shown here, like if this is a bacteria below here, a gram-positive bacteria, that you see that you have the membrane, which is the thin black line round, and then after the membrane, so that membrane is what you see as this bilayer. It's a lipid bilayer there, and then you do have the very thick black. That's a cell wall, which is made up of this peptidoglycan layer. Now, having seen this structure of the gram-positive bacteria, and we did talk about the synthesis of the peptidoglycan. Knowing the structure of that peptidoglycan, here we have represented it much more easier. So I mean, looking at it from the top down, that's why I've numbered it 1, 2, 3, 4, 5. What you need to look at is so you have this light, green portion representing the N-acetylglucosamine, and then a deep green is representing the N-acetylmuramic acid. So that's how they alternate. Then on the N-acetylmuramic acid residue, you have, the peptides, the tetrapeptides. It's formalized there. I think it's a pentapeptide, but of course, when they bind, you see that you have only 4 of those amino acids there. So say a tetrapeptide on this N-acetylmuramic acid. Now, look at this. You have this protein, which is being referred to as the penicillin binding protein. The penicillin binding protein, because that is the protein, or say the enzyme that penicillin targets. But that is if you are referring to it as, say, the particular enzyme that penicillin targets, then you call it that name. But if you are referring to its function, of course, then that protein, that enzyme is glycopeptide transpeptidase. So this will take you to the naming of enzymes. If you see a transpeptidase, what does it do? Quickly, you know that that's going to be a transferase. It might be doing some transfer between peptides. Glycopeptide. A glycopeptide is what? That is a carbohydrate with peptide together. So you see that the green and light green structure, which is composed of N-acetylmuramic acid, N-acetyl glucosamine, those are derivatives of monosaccharides. So those are carbohydrates on which you have peptides, proteins, that are bonded. Now we see that somewhere in the active site of that protein, that enzyme, is a reactive serine. I mean, that is being shown because we did look at mechanisms of action of enzymes in the last lecture. And we did look at what we refer to as serine proteases as one class of enzymes in which you have in the active site of these enzymes serine, the amino acid serine, that's having a hydroxyl group on it, and that hydroxyl group is reactive. Now quickly, what you notice is that in the active site of that penicillin binding protein, you have the serine that is interacting with the peptide. And so you see the first thing that happens is that you see, because in the bond you should have a tetrapeptide, but when you look closely at the peptides that are on them, they are 5. So those are pentapeptides. But the first thing is that one of the amino acids is removed. Alanine is cleaved off. So that's part of the peptide binds into the short groove. So if you see the active site, it's like an H, one short groove, one long groove, which are joined. You see in that active site, you have a second peptide, which is coming in. And in the end, you have, the yellow link, which is between those two amino acids, between alanine and lysine. So the keys are beside here. So you can look at it and step by step. If you see a red sphere, that is alanine. You see blue, that is glutamate or glutamic acid. If you see green, is lysine. And then the peptide linkage, which is formed between them, is a pentaglycine chain that is yellow. So if you can look at this key and see which color represents what. And you can easily understand that there is that, peptide linkage which is forming between alanine and glutamic, and lysine there. Now, this type of bond, this linkage, this cross linkage is going to be formed between these, adjacent peptides along the chain. And you see at the end, when you have the cross linkage formed, you have the glycopeptide transpeptidase dissociating from it because it's finished its work, it's gone, it can go and bind to others form another cross linkage. So the enzyme continues to form cross linkages at different portions. Once it finishes, it dissociates. All right, so this is a simplification of the process of the peptidoglycan synthesis. Now, we're saying that penicillin is a competitive inhibitor to the glycopeptide transpeptidase. Now, it is competing with what? It's competing with these, peptides, which we are referring to as the, D-alanine peptidoglycan ends. Because those are the ends. Remember, the red is alanine. So those are the ends that fit into the enzyme active site. So that's why it's being referred to as the alanine peptidoglycan ends. Which is to say that penicillin is having similar structure as that alanine peptidoglycan ends. In other words, penicillin is a structural analog of those peptides that get cross-linked in the biosynthesis of the peptidoglycan. And so you see, you have penicillin being represented with these structures, which are like, is it 6 or 9? But the moment that structure comes close to the penicillin binding protein, you have it opening up. So there's that reaction with it, with the serine residue in the active site of the enzyme. And penicillin opens up and fits into the active site like an H, fits in there. So it means that it's blocked. The active site is blocked. And once the active site is blocked, it means there cannot be synthesis of the peptidoglycan. If there is no synthesis of the peptidoglycan, then there will not be formation of the bacterial cell wall. In that case, it will result into cell death. Because of course, if the cell wall is not formed, then the bacteria is not protected. And now you see, if you look at the structures of the peptide ends, that alanine, ends that fit into the active site of penicillin, and you look at the structure of penicillin, you see that there is resemblance. They look alike, except that for penicillin, you see that there is a closed ring. These rings are closed, but when you look at the peptides, of course, these are peptides. You see, it does have, they are opened to fit into the active site. That's why you see that penicillin is shown to be having some ring, a close ring, that it is only when they come in close proximity to the enzyme, the penicillin binding protein or the glycopeptide transpeptidase, they open up and then they fit into the active site, blocking the enzyme from carrying out its function. Okay, so that said, we can even take a look at it much more this way. The diagram is just a repetition from the previous slide, but here you see that we're seeing it much more simpler, that you have a glycopeptide transpeptidase with a serine residue, and the serine residue is having a hydroxyl group in its side chain. Penicillin, as an antibiotic, comes and binds to that serine residue, forming, that penicillin enzyme complex, and renders the enzyme inactive. So all what you described simply, that's what it means, that you have penicillin binding to the active site of the glycopeptide transpeptidase and renders the enzyme inactive. And once the enzyme is inactive, you have the biosynthesis of the glycopeptide the glycopeptide being inhibited. And once that is inhibited, then of course, you cannot have the formation of the bacterial cell walls. All right. Now, when I think some of you who have perhaps done some industrial attachment in some pharmaceutical settings may have come across, this term, the beta-lactam antibiotics. It's been referred to a lot if you go to the pharmaceutical companies. Even if you just go to maybe a website of, say maybe Entrance Pharmaceuticals, any of these pharmaceutical companies which you have in Ghana, if you just go onto their website, you see they talk a lot about the drugs that they produce. I think some of it is beta-lactam antibiotics. And what is a beta-lactam? Is this ring? You see the ring we saw in penicillin that opens up to fit into the active site of the penicillin binding protein. That ring is a functional portion of these antibiotics. And so you see it in all the antibiotics. Now, these different types of rings, what is being referred to as the beta-lactam rings. So you see these ones, they have different names. So for instance, the first one is a pinam, second is a pinim. So if you compare pinam and pinim, the only thing is that you have a double bond in the pinim. So just like the way you say alkenes. So you have a pinam, you have pinim. And then the next one, you have a carbapenem. Why a carbapenem? You see, the pinam and the pinim, they're having sulfur there. In place of that sulfur, you have carbon. That's why I say a carbapenem. So it's just like the pinim, but the sulfur is replaced in there with carbon. Then you have a CFAM. So that's a 6-sided ring. It's no longer a 5-sided ring, which is fused with that 4-sided ring there. So you have a CFAM. And that CFAM, if you look at it, the next one is having a double bond there. So that's a CFIM. So you have these different rings which you find in these penicillin-like antibiotics, what is being referred to as the beta-lactam antibiotics. Of course, these rings are not exhaustive. There are many more different. But once you see this, the next time if you see a different one, you'll be able to quickly classify that particular antibiotic as a beta-lactam antibiotic. And on the Ghanaian market, we have this amoxicillin as one of these common antibiotics with a beta-lactam ring. So you see, the beta-lactam ring is there. That's amoxicillin. And you also have a common combination of these with clavulanic acid. So you see clavulanic acid there. What you do call amoxiclav. You see that. So it's a combination of amoxicillin and clavulanic acid, which is more said. That combination is done in order for the drug to deal with antibiotic resistance, because you have clavulanic acid being able to augment the function of amoxicillin. And even if you look at, so what's the ring? How will you classify the ring which is in amoxicillin, for instance? So if you look at a ring in amoxicillin, that will be the very first ring there, which is the pinam ring. That's it. Same, the pinam ring. Now about clavulanic acid. Clavulanic acid, you don't see, you see. So I said these rings are not exhaustive, and you could see that, yes, even the ring here in clavulanic acid, you don't find it as one of the examples up there. That ring in clavulanic acid is an oxypenam. So because you see that it's a penam, but in place of the sulfur, you have oxygen. So that is an oxypenam that you have there for clavulanic acid. So that's one practical application of this. Another thing is you have what we do refer to as prostaglandins, which are like natural analgesics. You can say natural painkillers. And these natural analgesics, are produced from arachidonic acid. That's a fatty acid. Arachidonic acid is a fatty acid with 20 carbons. And you have the enzymes phospholipase C, phospholipase A2, that will act on diacylglycerol or some phospholipid, and then and phospholipid, and together I have this arachidonic acid that can that will be produced. Either there's phospholipase C that cleaves off some phosphate group from phospholipid, and then you can have the production, and then there will be some cyclization reaction along the line, but let's not bother so much about that. What I want us to focus on is on these two enzymes here, COX-1, COX-2, which is cyclooxygenase. So these are cyclooxygenases. So these are some enzymes that catalyze some oxidative reaction in the biosynthesis of prostaglandins, and these prostaglandins, they act as I said, some painkillers, analgesics. As you see, they produce things like prostacyclin, thromboxane. These two, they do two opposite things. So, for instance, if there is inflammation in the body, you have the thromboxanes will be resulting into some coagulation, blood coagulation. They act in blood coagulation, whilst prostacyclins do the opposite. They rather improve blood flow, and so these two regulate the flow of blood to places of inflammation in the body. That way the pain that comes from inflammation is regulated. Now you have this. So these two, let's say the prostaglandins, they act in pain recognition. That's what it is. They will act in pain recognition, and which means that if you have the control of production of prostaglandins, then you can minimize pain recognition. So that way you can have an analgesic working for you. Now you see, you have the cyclooxygenases. These are enzymes which are responsible for the reaction in the conversion of arachidonic acid to these prostaglandins. And these will be the targets of some analgesics, common analgesics. So for instance, brufen, what you call brufen. Now brufen acts as an inhibitor to these cyclooxygenases. In fact, if again those who perhaps have done some things in pharmaceutical industries, maybe you've gone there for some industrial attachments, for instance, you would have come across, or maybe even if you just did work in a pharmacy, you would have come across some medicines with this that they have written on them, COX-1 or maybe COX-2, things like that. That is to say that the medicine actually targets those enzymes, becomes an inhibitor to those enzymes, cyclooxygenase 1 or cyclooxygenase 2. Now, from today, if you take any medicine and then you open the leaflet and you read, you should be having a better understanding of the things that are being written there. If you see something like a cyclooxygenase, you know that, this is an enzyme. So the medicine is going to act on this enzyme. Should be able to see these things when you read those things and understand them. And you have, of course, the substrate for that enzyme is arachidonic acid. The inhibitor, which is competing with the arachidonic acid, is brufen. So that is another common example as well. Another painkiller, aspirin. You see, aspirin also inhibits cyclooxygenase 1. That same reaction, the conversion of arachidonic acid to prostaglandin. So, and you see, you have this in the reaction will be occurring in these activated platelets. You know, when there's an inflammation, you then have some platelets that will be activated. If you remember, there's that reaction of platelets that will be involved in blood coagulation. That's a control of the inflammatory process, right? So that's another one. Aspirin acting as an inhibitor to that cyclooxygenase. That's what is similar to what we have just seen with brufen, that you have aspirin acting as an inhibitor to cyclooxygenase 1. Now, this is something which is applied for application in pesticides, like some insecticides. Again, if you take some insecticides and you look at the active ingredients there and you read, you see that those active ingredients are acting on the central nervous system, or let's just say the nervous system of the pest. A lot of times is to cause paralytic effect in the pest. So you have the enzyme acetylcholine esterase. Humans, we also have acetylcholine esterase, which is responsible for our muscle contraction and relaxation. You have the acetylcholine, which is like a neurotransmitter. It binds to muscle cells, and is binding to the muscle cell is a signal for the muscle to contract. And then when acetylcholine esterase comes to hydrolyze the acetylcholine from the muscle cell, then the muscle relaxes. So in every movement that you make, walking, running, talking, anything, your jaw is moving, all the movement, any movement that you are making, that your muscles are moving, you have this reaction of acetylcholine and acetylcholine esterase hydrolyzing acetylcholine from the muscle cells occurring. It's a fast reaction which is occurring. A lot of these acetylcholine esterase acting in muscle movement, muscle contraction, muscle relaxation. In the heart, as the heart is pumping, contracting, relaxation, there's a lot of this chemical reaction which is occurring. Acetylcholine esterase, acetylcholine, sending a signal, and then acetylcholine esterase going in to hydrolyze the acetylcholine, and then the muscle relaxes like that. It's a chemical reaction which is occurring that results into muscle contraction and relaxation. Now, that acetylcholine esterase in its active site is also serine, which means that acetylcholine esterase is also a serine protease. Now, you have the inhibitor diisopropyl fluorophosphate, simply referred to as DIFP. So you see, if you go to take the next time, just go take I say some mosquito spray, any of these pesticides you have at home or anything, take it and look for the active ingredients. And you see some of them are these, fluoride, fluorophosphates, things like that. You see a lot of them being written there. They go to bind. They go to bind to this enzyme, acetylcholine esterase, renders that enzyme inactive. And once that enzyme is inactive, it means that it will result into some paralytic effect in the insect. The pest will not be able to move. That's why you see that when some of this pesticide is being sprayed on some insect, maybe some cockroach or anything, and then you just see the cockroach is not able to move, or is at one place and turning around, and then it dies because it's not able to move. And at the same time, you have the enzymatic reaction, which is responsible for its muscle contraction relaxation. That paralytic effect is caused in the insect. And that also tells you that exposure to some of these insecticides can also have adverse effects for humans. Over a long period of time, you have these that can result into very serious problems. Sometimes immediately, it will not cause anything, but imagine that you have these pesticides which are lipophilic. They are lipophilic, so they dissolve in oil. And so when they are getting into the body, sometimes some of these pesticides get deposited in the adipose tissue that is under the skin. And it's as if they are not there. And so a lot of times in old age, some people suffer from some pesticide intoxication because they have been exposed to these things over a long time, and it accumulates. And in old age, when they begin to lose weight, and sometimes in some health conditions, when people begin to lose weight, it means that you have the adipose tissue under the skin beginning to break down. Then you have the release of these substances into the bloodstream in high amounts, and it begins to cause serious problems, which is why it's really, really important that you stay away from, many of these things. I do speak about this, the use of, the indiscriminate use of pesticides in Ghana a lot, where you have uneducated people like these farmers that they just go into some shop, and because there's not any proper regulation, and they go buy any of these things, mix it anyhow they want, and start spraying places. Even in the cities, people just go buy any of these things and spraying about the whole place, and people in neighborhoods get exposed to these things anyhow because of the loose regulation around and all of that. At home, we go buy any of these insecticides. The way the insecticides are being designed, when you use them, you get so much exposed to them. In other countries, these insecticides are engineered in such a way that you stay much away from them. Like, the way tear gas is done, so that you just open it, and then once you leave it, you sort of roll it into the room, you close the door behind you, and you leave. You leave home, come back hours later, and so you see, exposure is minimized. But if you come to Ghana, you see you are holding the can and you are spraying, and so you are being exposed to it more and more. And so you must be aware of some of these things and take precaution. All right, I think this is where I will stop talking and then open the floor. If you have any questions, you can raise your hand. I will enable your microphone. You speak, I'll respond. Alternatively, the chat is active, and so you can write in the chat. The floor is open.